I record, and this is chapter 14, right? Mendel and the gene idea. And this is the famous pea plant, right? That we're gonna talk about. Okay, so again, heredity. Heredity is passing inheritance to offspring, right? And some important terminology in genetics, like very, very basic terminology. Heritable feature is called a character, right? Like flower, color of the flower, shape of the seeds. Humans like eye color, ear shape, what else, like shape of the nose. So it's a character. Now, each character has different variations, right? For example, if you refer to the color of the flowers, it can be red, it can be blue, it can be yellow, right? Eye color could be blue, could be brown, could be hazel. So variation is called a trait, right? So for example, you have a red rose and you have a yellow rose, right? So they both roses. The color is characteristic. Yellow color is one trait, red color is another trait. Mendel spent some time in the university, and probably he got some interesting ideas that he was able to apply to practical, like he was able to apply it to field work. His field work was done in the monastery. I'm not sure, because he retired to monastery as a monk. I don't know how it's like exactly what was arrangement with their the management of the monastery about him being both monk and a scientist, but it definitely worked. And I think Mendel was lucky, not only because he definitely had a brilliant mind, but he was lucky because he had great patience. Sometimes in science, that's what actually matters. Matters to stick to your ideas, be patient, and kind of like, push hard. So Mendel selected for his first genetic experiments, he selected pea plants. Why peas? It's quite easy to grow them, right? Short generation time means like you can get results quite fast, right? Large numbers of offspring, which is important for statistics, right? And you can control the mating. Plants can be something called self-pollinating and cross-pollinating, and they can be both, right? So for pea plants, definitely can work both. So what he did actually to control whether it will be self-pollinating or cross-pollinating, you see, this is like in the plant, there's a stamen, right? With a carpel, this is the female part, right? So there's an egg cell, like maternal egg cell, female. Enters the it's like stamens with their anthers that have the pollen. The pollen is the sperm of the flower, right? So it can fall over the same flower's carpel, and it will be self-pollination. Or the wind can take it to plant nearby, and that will be cross-pollination. So what Mendel did to control it, he was cutting off the anthers, right? And he was brushing pollen from one flower to another to ensure, again, cross-pollination between different lines. So again, first of all, he created pure lines. What is pure line? Make sure that generation after generation, he was seeing the same trait, like color, right? You can select trait like color, you can select characteristic like color, you can select characteristic like color of the seeds, you can select characteristics like shape of the seed as well. So again, parental generation, that's basically generation that creates first offspring, F1. F for filial means like children. First filial generation offspring, F1, that he formed through their breeding. So again, at the beginning, he was tracking only one characteristic, right? Like a color. And to create, like to track it, as I said, he was doing pure or true breeding. So generation after generation, no mix. Okay. Then what he did, he took two true breeding varieties and crossed them, and that he called hybridization. Do I have a, this is the right picture, okay. So true breeding parents, purple and white, right? And first generation, F1, he got all flowers purple. So basically what happened, the white color disappeared in the first generation. And if Mendel will stop here and say, okay, you know, that's how it is. You cross purple and white and you get only purple. Done. The end of the story. However, again, he was smart. He was smart because he decided to take F1, the hybrids, and cross them, right? So two hybrids crossed. What he got a bunch of flowers. How many exactly? 705 plus 224, it was 929. So can you imagine like 929 flowers to count, right? To count and select them by color. And he found that, 705 were purple and 224 were white, which means that somehow the white color, the white trait didn't entirely disappear in hybrids, right? It was somewhere there, but showed up only in next generation, which is, again, that might be the reason sometimes certain traits, they skip generation, right? Something you see in grandparents and grandkids, but don't see in generation in between, right? So this is a summary. Let me just see if I share the sound with you. Share the sound.
In this recreation of Mendel's historic experiments, we'll have the opportunity to predict the outcome of two types of genetic crosses using a Punnett square. In this first example, the plants being mated differ in just one trait. One of the plants is homozygous for the dominant purple flower allele of the gene. The other plant is homozygous for a recessive white flower allele. To show the mating, we arrange the parent plants along two sides of the square. Note that each parent produces a single type of gamete. The genotype of each gamete produced by each parent, shown on the top, is brought down into each of the squares below it. Likewise, the genotype of each gamete produced by the other parent, shown on the left, is moved into each square to the right. The Punnett square helps us predict the genotypes of the offspring and their phenotypes. Notice that all these offspring have the same flower phenotype as their purple-flowered parental generation. In the offspring called the F1 generation, the white phenotype disappears. If we mate these heterozygous plants to each other, or allow them to self-pollinate, let's see what kinds of offspring will occur. Using the Punnett square, we can predict the genotypes of the offspring, and then the phenotypes as well. This first series of crosses recaps the results of a mating in which the parent plants differ in a single trait. The parent plants are homozygous for the dominant and recessive alleles. Their F1 offspring are heterozygous and show the dominant purple flower phenotype. The next generation, called the F2 generation, reveals a typical 3:1 ratio of phenotypes. The recessive white-flowered phenotype reappears in this generation after having disappeared in the F1 generation.
Okay, so we didn't talk about homozygous, heterozygous yet, but let's basically review this right now. Now, first of all, a gene, the word gene was introduced into genetics much, much later. So what actually Mendel called, he called it a heritable factor. So heritable factor that each parent contributes heritable factor that pretty much provides the trait to the offspring, right? And he was like, he was doing the crosses. As I said, he had huge patience, humongous, right? Because look at these numbers. Look at these numbers. So flower, seed color, seed shape, rod color, rod shape, flower position, stem length, right? And look, he was sitting and counting and counting and counting and counting, and pretty much all the time, the ratio was approximately 3:1, or 75% to 25%, right? So 75% that you're going to see the dominant trait, in 25% you're going to see recessive trait. But look at this, like counting 6,000, no, more than 6,000, 8,000, almost another 8,000 piece. Like again, counting with a yellow green color. So, but again, that was for the first time, as I said, first time scientific approach was applied to what humans were doing for, again, from the dawn of the history, selection. So okay, so this is our, this is a nice, but before, I'm gonna write something on the whiteboard. So Mendel called it heritable. He had no idea about chromosomes. He had no idea about DNA, right? This heritable factor we now call a gene. Now we know that half of genetic material we get from mother, half of genetic material we get from father, right? And we get half chromosomes from each parent. And each chromosome has its own pair, right? So if this chromosome from mother, this is chromosome from father, right? Let's say this is pair number 1. So each pair called homologous chromosomes. Homologous means they carry the same genes, right? So you can say genes are in charge of particular characteristic, right? So let's say characteristic could be, let's say we talk about, guys, can you help me Pick up a characteristic to discuss, please. Are you there with me?
Eyes,
What?
like blue eyes.
The eye color. Okay. Yeah, we can say the eye color, even though you're gonna see eye color is defined by many genes, but it's fine. Okay, so let's say eye color. Make things simple. And let's say the mother is brown and the father is so homologous chromosomes again means genes will sit exactly the same location, right? So usually this is called the shoulder. Whatever is above the centromere is called the shoulder. And usually this is called the short shoulder, this is called long shoulder. It doesn't really matter. What matters is that each gene has a locus, right? Locus is site of gene on chromosome. Now, uh, genes, basically one more time, responsible for characteristic, right? Trait is a variation of the characteristic. So variation of characteristic trait is called allele, okay? So allele is variation of trait or gene, right? So allele could be for brown color, allele could be for gray color. And if we talk about pea plants, right? So allele for purple, allele for white. So locus, locus, allele variation. Now, you can have different alleles, right? You can allele for blue, right? No, doesn't right. Okay, whatever. Okay, can be allele for blue, can be allele for brown, right? So if alleles are different, this condition called heterozygous. Okay, different alleles. Now, if alleles are identical, right, like purple, purple, white, white, this condition is called homo. Okay, identical alleles. Now, when it's homozygous and you're talking about one gene, it's pretty much straightforward, right? White, white, purple, purple. Now, if you have variations, right, then you can see the interplay. So if one is white and one is purple, right? But what you see, the color is purple. And there's another word actually for that. It's called phenotype. Phenotype is basically what you see, right? You can say what you see. You can say, gosh, I'm sorry. It doesn't work. Draw. Phenotype is actually called expression of genes. Expression again, gene can show up, right? Gene might not show up, right? Whatever shows up and whatever, like, again, whatever traits you see, that's the phenotype, right? And you can also say in phenotype reflects the genotype. Okay? Now, in case of heterozygous, right, if you have different alleles, what's going to be the phenotype? If you have different alleles and one allele takes over another, that's called, this allele is called, tell me. No? Are you there, guys? If you have allele for purple and allele for white, and the phenotype is purple, it means this allele is dominant or recessive?
Dominant.
Dominant, absolutely. So dominant. Dominant is one allele is needed for phenotype. In recessive, you need two, right? Two. So it's not like, again, that dominant suppresses the recessive. It doesn't work that way. It means that basically if you pair dominant with recessive, right, the dominant trait is the one that contributes to the phenotype. It doesn't mean that recessive, again, doesn't do much, but more contribution comes from the dominant allele than from the recessive. Okay, and one more time, I hope this material so far should be familiar to you, right? One more time, homologous chromosomes carry the same genes. Genes sit on the chromosome in what's called a locus. And if alleles are identical, that's condition called homozygous. Alleles are different, heterozygous. And then one allele can be dominant, one allele can be recessive, right? So what you can see here, two homologous chromosomes, two loci, right? Allele for purple, allele for white. And what you can see, dominant facilitates production of enzymes that probably in charge of synthesis of their color, right? And for the white, in this case, there is no enzyme. But again, doesn't mean one suppresses another one. It's just one is able to, again, to make a visible contribution while another one fails. In this case, so that's, I just explained to you dominant versus recessive. And what is law of segregation? So law of segregation means each has two alleles, right? I'm talking about the parent generation. But they can contribute only one to the offspring, right? Now the segregation is kind of random. And actually, in nowadays genetics, it's a lot about statistics, right? Do you like statistics, guys?
Yeah.
No, I see. Don't like statistics. Okay, so I mean, it's not a very hard one, to tell you the truth here. So, anyways, this is quite simple to demonstrate the law of segregation. Again, parental generation, they can contribute, let's say this one, purple contributes one of the purple alleles, the white contributes one of their white alleles. F1, they are hybrids, right? Purple and white. And usually in genetics, dominant allele goes as uppercase and recessive as lowercase, right? So hybrids, um, F1 generation can contribute either purple or white, right? Um, you know what? I think I'll better draw it just to make it easier. So parents, right? Purple, white. F1, purple and white. And F1, let's say this is one parent, right? So one parent can give one of the alleles. Another parent can give one of the alleles. So you get purple, you get purple, you get purple, and you get white. So you can see it's perfectly 3-2-1 in this Punnett square. So law of segregation means each parent can give one of the alleles. It's something that sounds like very straightforward to us, very much obvious, but actually it was quite a breakthrough, right? A breakthrough back then because it's for the first time explained why certain traits skip generations. Okay, guys, is it clear with this one, with the Punnett square? Any questions? How you get 3-1? Talk to me. Can I have some, like, any of the reactions, any of the responses that you are there listening? Please. Okay. Wow. It makes sense. Yeah, it makes a lot of sense actually. I mean, it's actually still, I believe it's still in use. Even like this one of the first of the simplest rules that Mendel proposed. Okay, so this I explained to you, homozygous, heterozygous as well. And again, phenotype versus genotype, right? As I said, phenotype reflects genotype. And I think actually a much better example of what is a phenotype? Cats. It's definitely more complex, but I think it's I love cats, so, but it can be any animal. So this is a good example of phenotype. Okay, cats are species, right? So definition of species is that they can mate within the same group, and that is called a reproductive barrier with other species. You can mate cats, for example, with their mountain lions, right? Even though they are relatives, right? Mountain lions and cats, but they're different species. However, no matter how different they look, here they can mate with each other, right? And they will form hybrids as well, right? If you take, for example, this is a Siberian, right? Beautiful Siberian cat. And I think this is a Macon, right? I think so. So Siberian Macons, they will mate and they will have their offspring, right? And they're going to have interesting genetic combination that you can see in the phenotype, right? This is phenotype of Siberian, this is phenotype of tabby cat. I think it's a tabby cat, right? This is a phenotype again. This is a Siamese. What is Siamese? I'm not sure. But guys, do you follow what I mean by phenotype? So by phenotype, you can tell, oh, this is a Siberian, and this is, for example, a Burmese. Yeah, make sense?
Yes.
Yes. The same you can you give an example with the dogs, the phenotype of the dogs? No, come on. There is a phenotype of a, let's say, Yorkie, yeah? Something that people like, right? What people like about Yorkies?
They're
They like
tiny.
it small, they fluffy, right?
Yes.
They, to my opinion, they're a bit too loud, right? But whatever. And what is the phenotype of, let's say, pit bull?
Big and muscular. Okay, I'm
Yeah,
not completely different. They're usually short-haired, by the way. So
yes. And they're a little bit mean. It also depends. So phenotype is not necessarily the looks. Phenotype can also be your personality as well. Yeah. So, but again, you select by phenotype. But as I said, phenotype is basically, let me go back. This phenotype, it means generations and generations of genes selected for this particular phenotype. So people selected for kind of like silver, for long silver fur, for fluffiness, right? For, I don't know, short ears. So these traits were picked up and ensured the next generation, right? They still the same species, but they have completely different phenotypes. Now, if you talk about one gene, it might sound kind of, again, much simpler and easier than when you talk about multiple genes that contribute to a particular trait. But what you can see here, purple phenotype can be homozygous dominant, could be heterozygous. For white color, it's always homozygous recessive, right? Okay, so sometimes you can say for sure, okay, if this is recessive, this is the phenotype and genotype. If you have heterozygous or homozygous for dominant, you might not be sure, okay? So if you want to ensure that whether you have homozygous or heterozygous, nowadays it's not a problem, you just run genetic like DNA test, but back then you could do something called test cross. So test cross is you take a phenotype that you want to figure out whether it's homozygous or heterozygous, and you breed it with the recessive phenotype. If you have dominant homozygous F1, they will be all purple, right? It's going to be hybrids. Hybrids that carry dominant allele. If it's heterozygous, then 50% will display dominant phenotype and 50% will display recessive phenotype. In one of the books, I saw like very nice picture. Instead of pea flowers, so crocodiles. Right? Do you know that crocodiles can be also albino? Have you heard about that? Guys, have you heard about albino crocodiles? Not that you ever cared, right? Yes?
yes.
So if you have a green crocodile and you wonder if it's dominant homozygous or heterozygous, you take this green crocodile and mate it with the albino crocodile. If first generation will be green, it's a dominant homozygous. If half crocodiles green, half white, it's a dominant heterozygous. Make sense? What is a test cross? Okay, and as I said, this is a monohybrids. Monohybrids, you basically heterozygous for one character, right? If you want to test, for example, for more than one allele, you can do it. That's called dihybrids, right? And this is dihybrid cross. Is it dihybrid or dihybrid? Guys, you say dihybrid or dihybrid? I'm not sure. Talk to me.
I think it could be said either way.
Yes, thank you, Dylan. Appreciate. Okay, I'll say dihybrid. It sounds more like I don't know, more like a Russian way to say it. So what Mendel did, he took not the flowers, he took the seeds, right? And seeds can have different characteristics. First of all, color and shape, right? So it can be yellow and can be green. It can be round and can be wrinkled. Now, at this point, Mendel knew that round and yellow is dominant and green and wrinkled is recessive, right? So he checked, this is for, this is the gametes from the P generation, yellow, round, wrinkled, green and wrinkled. So F1 generation, they all dihybrids for yellow and round. And in dihybrid, it's just you have more variations. So more variations in the segregation. So sperm and egg could have different sets of genes. So they can come as yellow round, yellow wrinkled, green round, green wrinkled. And what he saw in the phenotype he saw is pretty much the same. It's just, say like in a square, right? So the ratio was 9 to 3 to 3 to 1. So 9 display dominant phenotype to 3 and 3, which is kind of intermediate, and 1 is recessive, right? What Mendel basically figured out from this E-hybrid cross, he figured out the law of independent assortment. Independent assortment means these genes for color and genes for shape not in any way actually affect each other, right? They don't go together, they go independently, right? So independently means you can have a yellow color and wrinkled shape. You can have green color and round shape, right? So regardless of parental generation, regardless of F1 generation, in F2, you see combinations that you don't see in parental and in F1 generation. And these combinations, as I mentioned, yellow color, wrinkled shape, green color, and smooth shape, right, round shape, they are due to independent assortment of alleles in the gametes that parents contribute. Okay, you're gonna see later that it's not really applicable to all genes. Remember crossing over, right? Remember crossing over. Mendel didn't know about crossing over. He didn't know that in gametes, genes kind of like blend, right? They blend from parental genetic material, right? The further away genes located from each other, if they located on different chromosomes, that's 100% true. if genes located are very, very close on the same chromosomes, they have more chances to go together. So independent assortment has some exceptions in that rule, depending on the position of the genes on the chromosomes. So you can see applies only to genes on different non-homologous chromosomes, those far apart on the same chromosome. Okay, and then it goes with the probabilities. So this is statistics. Guys, how are you so far? My most quiet class this semester. Okay, good. Even though you're quiet, I still enjoy teaching you. Okay, so this is addition row. Okay, so segregation and addition row. And this is about probabilities, right? So can anyone tell me what is a probability? D1, what is probability?
It's basically just a chance of something happen.
Exactly,
Yeah, I agree
right?
with you, man.
Yes, chance something happens, and you can actually calculate these chances, right?
Professor, it's like the part you were saying statistics, right? Where it's like the rate and the proportion basically of how likely is something to happen, correct?
Exactly. So, before we're gonna continue with some math and statistics, let me just again demonstrate like Punnett square. So, there's some forms of like there's certain cases of deafness. They come as genetic, right? So let's say parents. This one. So D. So D is sometimes with this pen. So D is normal hearing. And D is. So let's say you have a couple and they know that some family history of deafness. And happens that mother and father, they both carry allele for hereditary deafness. They're not affected, but they carry. They call carriers, right? Genetic carrier means you have the allele that doesn't affect your phenotype, but you can pass it to your offspring, right? So they both carriers and independent assortment. Father can give one of the alleles and mother can give one of the alleles, right? No deafness, carrier, right? So you can see 3, 2, 1. So, and probability is 25%, right? That the child will be deaf. And my question to you, is it a high probability, 25%? Is it, to your opinion?
Could you repeat what you said, Professor?
I'm saying that probability that the child will be deaf is 25%, right? And my question to you, is it a high probability to your opinion? 25%?
Compared to the rest of it, no, because it's a 1 out of 4 chance.
I should say 25% is still decent, right? It's still decent probability that the child will be affected, right? And that's what basically they're gonna probably hear in the genetic consultation if they go. So you take a chance, right? And the question is, let's say if they had 3 children, right? and all three children were deaf. Do they have high chance with the fourth child, or is it going to be the same?
Will it still be the same?
Yeah, it's gonna be.
It will be the same. Doesn't matter how many children deaf, how many children hearing, every time it's gonna still be the same 25% chances. So that's what, again, you apply to genetics, right? So you look at this one, it's like tossing a coin, right? So that's about segregation. So parents are hybrids, right? So you toss a coin that in half, like 50% chances you give dominant, 50% chances you give recessive, and the same here, right? And then you calculate that it's gonna be 1/4, 1/4, 1/4, right? So 25% chances dominant homozygous. You see 50% chances will be heterozygous, and 25% chances it will be recessive homozygous. Okay. I should say when you talk about monohybrids, it's definitely 25% each one, right? Or like 50%, half, right? Half chances of carrying dominant, half carrying recessive. When you go to dehybrids or more, it's getting more and more complicated, right? So addition rule is that when you add, again, different probabilities, right? So you add here and here because they are identical. So this is different, right? This is 25%, but this is 25 and 25, you can add. So you have 50% chances having heterozygous, 25% homozygous, 25% homozygous. And as I said to you, it's getting a bit more complicated when you look at the dehybrids, right? So with dehybrids, you see you multiply, right? For this genotype, right? With the genotype, you multiply 4th over half, right? And this is, again, probability of their gametes occurring together. But you know what? What time is it? It's 9:40. Let's take a 10 minutes break and we're gonna talk more about types of dominance, which is actually more interesting than statistics and probabilities. Okay, guys, I'll see you in 10 minutes.
Okay.
Okay.
Let me resume recording and let's continue. So single gene, right? Single gene is a very rare thing. Good example, like, of single gene inheritance in humans is if you can fold your tongue. I can't, right? If I try hard, I can't fold my tongue that it look like a tube. Can you do it, guys? Like, how many of you? You don't have to show it to me, but if you can do it, just thumb up. Should be more. Guys, should be more. Yeah, if you can fold your tongue, it's a dominant trait. If you can't, it's a recessive. But it's a good example of one gene, right? Now, I have no clue what's the advantage of being able to fold your tongue. Can you think about it? Because usually traits that they beneficial, they usually go as dominant. So what do you think? What's beneficial in being able to fold your tongue?
I mean, I don't know about folding like benefits of folding your tongue, but I know there is one that you can move your ears, and it's just like for helping you hear something better, like picking it up, like animals do it, like rabbits or
Yes,
something.
like tuning your ears. Yeah.
Hmm.
Mm-hmm. But yes, I mean, I should say humans accumulated so much of genetic material that sometimes it's really hard to think what is actually for. Yeah. Like, as gonna be another example, like widow's peak, right? Widow's peak, this is like the hairline. So why some people have this hairline versus another one? Not clear as well. I know, for example, like with the eye color, but it's not the single gene inheritance. With the eye color is definitely defined by presence of melanin, their pigment. Like no melanin is gray, right? A little bit is green, a little bit more blue. You add more like hazel and then brown and dark brown, right? So it's kind of like, but so many genes control how much melanin is actually deposited into the eye that um, it's as I said, it's like it's a complex inheritance. But the bottom line is just how much pigment uh, is accumulated in the iris, in the colored portion of the eye. Okay, so extending Mendelian genetics for single gene. Okay, so complete dominance is one phenotype is basically takes over completely, and when both dominant homozygous and heterozygous are identical, like with the flowers. So purple is completely dominant over white. Incomplete dominance is when hybrid is kind of like a blend, right? In between. Codominance is when two dominant alleles actually contribute to phenotype. So you're going to see it right now. This is example of incomplete dominance. Incomplete dominance, red, white, and hybrids are pink, right? So 50% chance pink, 25% red, 25% white. So as I said, as if you just took two colors and mixed them together, red and white give you pink. Codominance, I think we're gonna talk about this later. The most common example of codominance is actually blood typing, right? And for example, could be allele for blood type A, could be allele for blood type B, and there is AB blood type when both alleles actually are expressed. Okay, that's for codominance, but I think we see it more. Okay, and we just talked about their color like that is pretty much contributed by certain enzymes, right? So dominant allele codes for enzymes that gives one form, and recessive alleles codes for enzyme, which actually leads to different accumulation distribution, right? So for example, like brown seed versus wrinkled seed. And Tay-Sachs, actually, it's a recessively inherited disease, right? But what happens actually that if you have recessive gene, if you're a carrier, right, it's incompletely dominant, right? But it's incompletely dominant on the biochemical level, okay? At molecular level, alleles are codominant, which is basically indicates like both examples, right? Of incomplete dominance and codominance. And frequency of dominant alleles, it's really depends, okay? Polydactyly. Polydactyly is a condition when there are extra fingers or extra toes, right? It's a dominantly inherited condition, but even though it's dominantly inherited, it's extremely rare because of the frequency of the allele, right? So, for example, I don't know, have you seen like people that have dark hair, but kind of like blonde or brown eyebrows and beard? Have you seen that?
Yes,
Yeah.
I've seen that.
Yes, it's also dominant inheritance, right? But it's rare because the allele is very rare. Okay, so I mean, it's if it's dominant, it doesn't mean right away it's common, right? It only means that it's dominant. I had like a long time ago, I had a presentation PowerPoint for genetics class, not for BIO210, it was just for general biology. And I put a picture, you know, there beginning of the presentation. So it's like genetics, and the picture I put it was actually polydactyly. So the hand that on their thumb there was extra finger, right, like tiny finger. So when I turned it on in the class, one of the students actually raised his hand and said, "Look, it's just like me." I felt so embarrassed to tell you the truth. First thing I did, I just removed that picture from the presentation. So, I mean, again, what are the chances of talking about genetics, having a picture in the class, and having a student with a condition? It's really, I should say, like probability is very low, right? Um, more common actually is having extra toes. Have you ever seen extra toes rather than extra fingers?
No,
No.
no.
Another common condition is that when toes are fused, like there's like a skin flap between the toes, or even like between two toes only. It's also actually dominant inheritance. Okay, so this is multiple alleles, and this is for blood type. Um, do you know your blood type, guys? Do you?
I don't know mine.
You know yours?
I don't.
No? It actually can come quite handy to know your blood type.
Yeah.
Okay, so you know about blood typing? I can explain like from the very beginning. Okay, so first of all, why should you know your blood type? Why it's important?
In case you need a blood transfusion, I know that you have to have a certain type of blood, um, to
Exactly.
to uh, to go along with the other person's blood. So it's not a basically so your body doesn't attack the new blood cells. So
Yep. The thing is that when you talk about blood type, you talk about the red blood cells, right? So the red blood cells, or they also called erythrocytes, are the most numerous cells in the blood, right? That's why our blood is red, because we have red blood type, red blood cells. If we had green blood cells or yellow blood cells or silver blood cells, our blood will be different color. By the way, why the color is red? Because of what element? What? What element, what chemical element gives red color to the blood? Iron. If you have, for example, copper binding oxygen, our blood will be purple, like octopus. Okay, so red blood cells, they cells, right? So being a cell, you have a cell membrane, right? Remember cell membrane? What you have on cell membranes, you have proteins. And we talked about different types of proteins that could be present in the cell membrane. And one of the proteins serves as their antigen. Antigen is a protein that basically cooperates with your immune system, right? As long as the immune system recognizes this antigen, right, it will never attack your cells, right? So antigen is like ID molecule. It's something that if you remember going to BMCC, what did you have to show at the entrance? No, tell me. What did you have to show in the entrance? Guys, talk to me. What did you have to show in the entrance to get to BMCC? Your
Yeah, student ID.
ID. Do you still have your students' IDs?
Okay, I'm sorry. I've never been to the campus.
Are you never been to the campus?
No.
Oh,
No, I haven't either.
yes, yes. So you have to show your student ID, right? If you show your, you show different ID, they won't let you in, right? So antigen is like ID. Now, antigens can have variations, right? So if you have certain protein A, right? So you have antigen for A. You have a certain protein B, you have antigen for B. You can have both A and B. You have none, right? So you have none, it's 0 or O, right? But the thing is that antigens come with a package, right? In your blood, you have molecules produced by immune cells, and this molecule is called antibodies. Antibodies usually depicted as like tiny forks, like Y-shaped protein. And the rule is that if you have antigen for A in your blood, you're going to find antibodies for B. If you have antigen for B, you have antibodies for A. Interestingly, if you have AB, you have no antibodies. And very, very interesting, if you O, you have antibodies for both A and antibodies for B. Now what's the problem? The problem, like no problem, as long as you don't get your, like, as long as you don't get a blood transfusion, right? The problem during blood transfusion, if you get a wrong blood type, let's say if you O, right, and you have antibodies for A and antibodies for B, and let's say you get blood type A instead of O, so you have a bunch of red blood cells will have antigen for A. So this is yours and this is not yours, right? Now your antibodies gonna bind. They bind like key and lock, right? So they match. So when antibodies bind to other red blood cells, what happens? They form complexes. They start to stick to each other. Antibodies stick to red blood cells and stick to each other. So what's gonna happen? This all red blood cells and antibodies and whatever gonna form like kind of like heavy chains. These heavy chains will sink in the blood vessels and clog circulation. So they're gonna clog circulation and the person will die. It's actually very hard, like heartbreaking death. When a person gets wrong blood transfusion, they don't die right away. They die within like a few days from actually from blocked circulation. Just 1 second, guys. Give me 1 second. I'm sorry. Okay, so this is called antibody antigen reaction. Now, as I said, so this is the genotype, right? O, this is genotype A, B, and A, right? And you can see like triangular is there antigen for A, circle is antigen for B, and this is AB. And one last thing, if I go back to my table, let me explain you something. So let's see, this is blood type, right? O, A, B, AB. This is antigen, and this is antibody. So O antigen is no antigens, antibodies for A and B, antigen for A, antibody for B, antigen for B, antibody for A, antigen for A and B, and no antibodies. So which one is universal donor? What do you think? Which blood type can actually go to other blood types?
O.
Oh, absolutely. It's universal donor. But O can receive only
O.
O, right? A can receive O and A, right?
Yeah.
And it can go to A and AB, right? B can go to B and AB and receive O or
B.
B. AB can go to
Um.
AB can go to
Only A.
AB only, right? But it can receive
Everyone. So it's the universal
universal
giver.
recipient, right? So which means that if you O, you are in, let's say, your situation is less fortunate, from example, from AB, right? Because AB can receive anything. But this is the most rare blood type, AB. On top of this, on top of AB, there is something called RH. Have you heard about RH? Have you heard about positive, negative?
Yes, I have.
Yeah. Guys, can you also talk, not only Jamaris? Can other people talk as well to me, please? Okay, so anyone here who is RH negative? If you know. 85% are RH positive. Only 15% are RH negative. Okay, so the thing is that it doesn't matter if you what your blood type. If you have additional antigen RH, means you are positive. RH is from the word rhesus because for the first time they were found in monkeys, rhesus monkeys, right? So if you are positive, can you get blood from negative? Yes. If you're Rh positive, can you get blood transfusion from somebody who's negative?
No, you can't.
You can. Doesn't matter. If you're positive, doesn't matter. Get positive or negative. If you're negative, then you can get only negative. Okay. So again, usually it's not a problem, right? So when you get blood transfusion, again, most people positive, so it's not a big deal, right? You should really watch out if you are negative in case again you need blood transfusion. But the biggest problem was, and it's not really a problem nowadays, is conflict between mother and the fetus. Have you heard about that? Anyone? You are Rh negative mother. you might have problem. Have you heard about that, guys? Don't just say yes, no, like come in. Guys, say yes, no, right, yes, no, if you ever heard about this condition. Okay, Jamarius, you answer. If you ever heard, if you can answer yes or no, fine with me.
No. A lot
No.
of people in the chat said no.
Okay. So, now I should say listen to me because your next quiz is gonna have a question on that. Can I have your attention now? Can I have your attention now? Yes? You're gonna have a question on what I'm gonna explain right now about RH conflict. Okay. Okay. So RH conflict is, let's say, mother, right? So mother is RH negative. Father is RH positive. Now this is a dominant gene, so baby will be very high chance baby will be RH positive. So during first pregnancy, everything goes well, right? But when woman delivers a baby, what happens usually? The blood mixes. The maternal and fetal blood get mixed during delivery. And after delivery, mother will produce antibodies for RH. So in her blood, she's gonna generate antibodies against RH antigen. I think I should have a picture. You know what? Give me 1 second. I'll stop share for a second. I think. Okay, wait a minute. Where did I? I think I closed internet. I don't know how to open it. Okay, second. Sorry, I. This is better. Okay, so I see RH incompatibility. So as I said, during first pregnancy, usually everything goes well. After delivery, you see the green antibodies, right? Now, next time she's pregnant again with RH positive child, this antibodies will cross placenta and will attack their embryo's red blood cells. Okay, so without red blood cells, that's pretty much certain death. So if baby won't be stillborn, baby will be greatly affected, will be very, very sick at birth, right? For centuries, women who were Rh negative, they couldn't have more than one child. So all the following children were stillborn. Nowadays, there is a medication actually that blocks them from, like prevents them from making this antibodies. Okay, so nowadays they can have as many as they want, right? But just be aware because there are a few cases I read that when they didn't know about this. It's called a rhesus conflict between their mother and the embryo. But I read about that. Can you believe? Doctors actually did blood transfusion for the fetus inside the womb, okay, to prevent the destruction of the red blood cells. But sometimes what happens actually, if not for the first child, but let's say for following children, even having different blood type between mother and embryo can have some effect. Sometimes there is, again, some conflict between different blood types. Okay, any questions here? Talk to me.
No, not really. I personally don't have any.
Okay, is everything clear, guys?
Yes.
Okay, so pleiotropy. Pleiotropy is when one allele is responsible for multiple symptoms. Good example will be sickle cell disease, when actually one allele that codes for kind of like wrong shape of hemoglobin has a profound effect on health. Okay, so one allele, many traits. Or polygenic inheritance, exactly opposite. Okay, when one trait is determined by many genes, right? Like height, eye color, skin color is a polygenic trait. And there's something interesting, very interesting condition called epistasis. What is epistasis? Epistasis, you have two different genes, right? But one gene affects expression of another one, okay? And here's example of Labrador fur, right? Labradors can be black, which is dominant, or can be brown, which is recessive, right? But there's another gene, let's say E, which actually controls deposition of pigment in the hair, right? So if you have dominant, your hair will have color. If you have recessive, the hair won't have any color, whether you have black or brown. So look at this, okay? Recessive, you see, recessive, no color. So if you have dominant black, dominant black, dominant black, dominant brown, right? But again, doesn't matter, brown or black, if you have recessive form of allele that controls the position of the color, it will have no color. That's called epistasis, okay? So when one gene controls expression of another gene. Is it clear, guys? Guys, is it clear?
Yes.
Okay, so Jamaris, you're responsible for the whole class. Really cool. Okay, and another one is what called quantitative characteristics. Quantitative is exactly what I just told you about the eye color. Now tell me, like I want to see how, oops, sorry, how will you listen to me? So I told you their distribution of their pigment in the eye. So when there is no pigment, what will be the color? What will be the eye color if there is no pigment? Oh my gosh, that's how you listen to me. What will be the eye color if it will be a little bit of pigment?
I guess something around blue, maybe dark blue.
You want no color, that's gray. A little bit color is green. More is blue. And the most is brown. Okay, so this is actually quantitative variation. It quantitative is means that like how much um, you see something called additive effect. Additive effect like contributes to spectrum, right? Like for example, skin color, right? So skin color is how many alleles you got that contribute to expression of melanin. The more alleles you accumulate, the darker will be the skin. The less alleles you accumulate, the lighter will be the skin. The same goes for the eye color. The more you accumulate, darker the eye, the less you accumulate will be gray. Okay, and as I said, the same additive effects go for height, for hair color as well. So this is a polygenic trait, but it's also, you see, environmental impact. So environment definitely has a huge impact on the phenotype, right? Especially nowadays, you can do so many manipulations with your phenotype, changing your hair color, changing your eye color, changing your body, right? Plastic surgery, so changing, like, yeah, a lot of things, just pay. But at the same time, you see, this is quite interesting. So this is the flowers, hydrangeas. If they grow in the basic soil, red. Some aluminum contributes to color of the flowers. So you see, it really depends. It depends whether you plant or you not plant, and depends where you grow, right? So, which means again, genes have huge impact, but genes is never 100%. So there are still some environmental effects. Okay, so any questions so far? So we pretty much like, again, we are done with our like basics of inheritance. Now I'm gonna talk about some genetic diseases. Are you with me, guys? One more time before I continue.
Yes, Professor.
Yeah, please, reaction. Yeah, can I see reactions? Okay, not bad. Not bad, I should say. Okay, pedigree. So, do you have your family trees? Anyone?
Yes.
Yeah. So, I mean, family tree can be quite handy in case, again, you're interested in genetics. But I mean, in our days, it's again, it's a matter of genetic testing. But back then, pedigree were actually something that you do some genetic research on. Just 1 second. Okay, so for example, to answer the question, what nature of inheritance, dominant or recessive, right? And I already mentioned the widow's peak, right? And you can see that if you look at this pedigree, right? First generation grandparents, you see in every generation. So what do you think? Is it recessive or dominant? If you see in every generation?
Dominant.
Dominant.
Absolutely dominant, right? Another one, quite interesting trait, is PDC. If you had like a real lab, you probably will try it as well. PTC is a chemical that tastes yucky, and not everyone can taste it. So usually in the lab, they have like strips of paper so students can taste. And they say, "Bleh," means they can, and they say, doesn't taste like anything, you can taste PDC. Okay, I forgot what PDC stands for, but it's just really name of the chemical. So you can see this is a dominant recessive inheritance, right? So carrier, a lot of people carriers, actually. It's not that uncommon gene, but even you carrier, you can't taste it, right? Carrier, recessive. So this is recessive inheritance. So let's
During the 19th century, the Austrian monk Gregor Mendel performed specific crosses with pea plants and worked out much of what we know today about the laws of heredity.
just 1 second, guys.
During the 19th century, the Austrian monk Gregor Mendel performed specific crosses with pea plants and worked out much of what we know today about the laws of heredity. We will use his ideas to consider the inheritance of a single trait, freckles. The inheritance of freckles is determined by a dominant allele. Suppose a freckled female who was homozygous dominant, big F, mated with a homozygous recessive male with no freckles, little f. How likely is it that their child will have freckles? A Punnett square helps determine the probable outcome of the genetic cross. Both parents are homozygous, so each can produce only one type of gamete as far as freckles are concerned. The female produces gametes with the dominant allele, big F, and the male produces gametes with the recessive allele, little f. Each square in the resulting table is filled in by combining the alleles from the sperm and eggs. In this example, all the children would be heterozygous for the trait big F, little f, and therefore would have freckles. Now let's consider why it's possible for parents who are both heterozygous for the freckle trait to have a child without freckles. The dominant F and recessive F alleles segregate during meiosis, so half the gametes bear the dominant F allele and half bear the recessive F allele. Filling the boxes, we see that the probable genotypes of the children would be in a genotypic ratio of 1 homozygous dominant, big F, to 2 heterozygous, big F, little f, to 1 homozygous recessive, little f. Phenotypically, the children would be 3 freckled to 1 not freckled. A child from these parents would have a 25% chance of not having freckles.
Okay. So as I said, genetically, in most of genetic diseases have recessive inheritance, right? So recessive inheritance, which means it's rare, right? And parents have to be carriers. I just explained to you. So parents have to carry recessive allele while they are not affected. So like albinism, right? Albinism is actually when cells in skin called melanocytes don't produce pigment called melanin. So if both parents are carriers, again, 25% chances child will be affected, will be albino. Again, albinism has quite a range, right? The problem is usually, you see that's called, I didn't even know this word, consanguineous mating is actually mating between relatives like cousins. Increased chances that as relatives can both be carriers, so increased chances of children having genetic disease, again, even if it's rare. That's why, again, a lot of cultures, they frown upon actually having marriages within families. One of the most common is cystic fibrosis. Have you heard about cystic fibrosis? Any one of you?
No, I don't think I have.
Okay, very likely that you do. As usually during pregnancy, they test for cystic fibrosis. It's better again to be aware if you a carrier. It's actually affects gene that codes for chloride transport channel. Give me 1 second. I'm sorry, guys. Just 1 second. Sorry. Okay. Chloride transport channel, which actually sends the chloride ions outside the cell and fluids follow. When fluids follow, that causes accumulation of mucus in different parts of the body. It's also actually causes, makes like a skin kind of salty because of the chlorine ions as well. We normally produce mucus in our airways, digestive tract, urinary tract, but in case of cystic fibrosis, there's overproduction. This overproduction clogs airways. It causes sterility because it also blocks reproductive tract. When mucus builds up in the airways, it usually complicates with pneumonia, all sorts of infections. For a very long time, those who had cystic fibrosis, they couldn't live like older than teenage years. Nowadays, they live much longer, but they have to go to the hospital from time to time to have their airways declogged, and they have to take like 30 pills a day. Can you imagine like 30 pills just to control this condition? A lot of research is going on. A lot of families, they have fundraisers and accumulated millions of dollars to do genetic, how to say, like genetic engineering, or like change the mutation inside the cells. So I didn't check much lately on their success, but I know that a lots of going on right now for cystic fibrosis. Okay. Sickle cell disease. I'm sure you're familiar with sickle cell, right, guys?
Yes.
Yeah. Sickle cell is not that, it's not that uncommon, right? Why it's not that uncommon? Because, carriers are protected from malaria, right? If you're a carrier, if you had a zygote, in malaria, it's a common infection, and I should say until now, it's treatable, but until now, it's even with the treatment, it has a profound effect over health, right? So for this reason, being protected from malaria, this mutation actually was favored from generation to generation. That's why it's so common. So you see, this is a carrier, right? So carriers have both sickle and normal blood cells, and that's, as I said, that's protective. If you affected, your cells have a sickle shape, can carry oxygen. The problem is the cells are very, very rigid. They cut off circulation around the body, which contributes to pain, fever, and organ damage. So that was about recessive. So recessive, as I said, 25% chance if both parents are carriers. Less common, dominantly inherited disorders, but in this case, child has 50% chances to be affected, right? Achondroplasia, this is a form of dwarfism. It's actually a mutation that controls the growth of the long bones. So other bones not much affected, but long bones in the arms and legs become too short, right? So if you see a parent, their achondroplasia phenotype, right? Heterozygous, normal phenotype, homozygous, then you can see 50% chances that children will be affected in this case. Another very common, not that common, have you ever heard about Huntington disease? You say yes, no, have you ever heard about it?
No.
Okay, good for you. It's a neurodegenerative disease, and when basically when gene kicks in like late 30s, 40s, and it has a devastating effect over the nervous system that starts with personality changes, movement disorders, and ends up in death. But movement disorders, that's what's called a Huntington's chorea. Chorea means uncontrollable movement. So people are in constant motion. Constant motion, they can't stop it, they can't control it. It just breaks them down because people burn so many calories and they can't even eat because of this like uncontrolled movement. What's the catch with this disease? The catch is that you see no phenotypes until age 35. So these people, without knowing about their condition, they can marry, have children, right, and pass the gene to the next generation. So again, fortunately, it's rare. Unfortunately, there is no cure, and unfortunately, you can't prevent actually. You can't control having these genes in the next generation. Okay, and multifactorial. So what are multifactorial? Multifactorial means they don't have clear genetic background. Like, I mean, their genes, no, they do have. I mean, genes increase chance, increase risk, but they don't, I mean, nothing more than that. So, for example, breast cancer. If you ever heard about BRCA2, so if you inherited these genes, they give you high risk for breast cancer. But I mean, like if you inherited cystic fibrosis, it means you have cystic fibrosis. There's no such a thing as you just have a high chance. Okay, but for certain type of cancer, diabetes, mental illnesses, right? It's like a combination between inherited genes and environment. Okay, so it increases your chance, but it's not the destiny. Okay, you understand? Guys, like 5 more minutes, please, like answer me when I ask you questions. Okay, thank you, Iwana. And genetic counseling. So again, if you like, let's say sometimes they do just if the mother is older than 40 years, or again, if family turns for, they want to figure out what chances the child will inherit. But, and you see, it's based on Mendelian genetics and probability. So the answer will always be chance, right? Because remember, independent assortment and rule of segregation, it's all about chances. And fetal testing. So I don't know if you heard about this. Fetal testing, not necessarily for genetic testing, sometimes just to like karyotyping, just see if condition like Down syndrome or so. I know nowadays I think there's a blood test that we can check for this without actually having having this procedure. But again, after age, let's say 36, 37, women have high chance to have child with Down syndrome, right? So sometimes what they do, they with a syringe, they kind of like go through their belly, collect amniotic fluids, and these fluids usually have fetal cells, right? So they grow cells on the dish, they bring cells into division. Remember something we talked about last week? And then they take a picture of the chromosomes, and computer actually will arrange them. And you can see if it's a boy or girl, and you can also count the number of the chromosomes. So this, like for karyotype, sometimes for more precise genetic testing, they actually take the chorionic villi. Chorion is one of their, it's one of the membranes around the fetus, and it has fetal cells only, right? So you can have a bigger sample, and this they can run DNA test and test for for certain genes.
So I have a quick question.
Yes, Jamarius.
Why is it that after a certain age, women are like if they get pregnant, they're most likely to have a child with that Down syndrome? Like, why is that the case?
You know, it's really interesting. I recently read an article which was actually about all sorts of genetic disorders, like how maternal age. It's actually very young age and older age. It's just the thing that not too many girls have children like at age 16, 17. But interestingly, at age 16, 17, there's also a very high chance for genetic mutations. So, which means that there is a age like window, like biological window to have children, let's say between age 20 and let's say 35. It doesn't mean you can have a healthy child when you're older, right, or younger, but the risk is much higher. Um, it's
So when you're younger, it's the same thing also?
yeah, but very young. Again, as I said, we don't really see it because not too many young girls have children, right? So it was always believed that it's because of the older age, but it's actually, as I said, it's like our like genetic, no, sorry, it's like statistical like distribution, right? So actually having a healthy egg cell is a big deal, surprisingly. It's really, really big deal. Most of the egg cells are actually not good. So um, I think people will appreciate having a healthy child. It's actually, it was always a miracle. There's so
Wow.
many eggs that are discarded because of some genetic issues, chromosomal issues, usually chromosomal issues. And there's so many fetuses that don't develop because of the same problem.
Well, thank you for answering my
Yes, it's let's say girls born with like maybe like 250,000 egg cells, and maybe like 200 of them are good for fertilization. Really good. Good enough. Okay, newborn screening. So newborn screening is actually certain conditions might be not apparent with genetic testing, but could be apparent at birth. So they usually check newborns for a condition called phenylketonuria, or PKU. It's also recessive condition, and that's when babies can metabolize one of the amino acids. If you take this amino acid out of the diet, they will be fine. But if you're not aware, it can really affect development. Um, they also test for thyroid condition. This thyroid gland produces very important hormones. If they're missing at birth, it can also compromise health greatly. Okay, so this is it, guys. I stop